Thanks, Diane. Thank you very much for the opportunity to chat to everyone about the virtual eye. And what I really want to do is to share my enthusiasm for mechanistic computational models of the eye, and then fairly swiftly um, go through three things. One, the broader context of multi-scale modeling of biological systems. Then, particularly given this group, look a little bit at metabolic modeling, and then finish up with some thoughts about blood flow. I'm trying to give a broad coverage, and so the tendency will be to signpost to publish work. And I'm going to focus on the retina, not because the rest of the eye is insignificant, but again, I needed to constrain things a little bit. So disclosures, I'm not a biochemist. I'm not a molecular biologist. I'm not a mathematician. I'm not a computer scientist. So a lot of the nitty gritty of this, I can't do. But a long time ago, as a pathologist, I became intrigued by how on earth do all the different cell types in a tissue interact with one another. And then when we impose disease on top of that, how do the adaptations that all of those different cell types make kind of act themselves out? And it seemed to me that actually, although it's fun to make up lots of stories, the interactions are so complicated that maybe we need to model. And I think the main motivation, at least from my perspective, around computational modeling is to try and understand how things work. And in particular, as I said, how they fit together, how are they balanced and optimized. Probably won't be time to go into this, but I'm very interested in how heterogeneity is accommodated. And then, of course, there's a whole raft of issues that relate to how it goes wrong. And then, as I've mentioned, when it goes wrong, how does it adapt? How does it adapt in aging? And of course, the clinical imperative is if it's broken or on its way to being broken, how do we fix it, both at the group or population level and for individuals? And the kind of individual strand in this collides with the really quite impressive work now that's being done around digital twins for healthcare. Digital twins have existed in engineering and the built environment for some time, but now people are really feeling that we can get traction on how individuals can have computational models of organs themselves. Peter Coveney and Roger Highfield, who are pictured in the bottom left-hand corner here, giving an interview, have produced a popular science book called Virtual You, which is quite a fun read. And this is just a random collection of things I pulled off the internet. But the area where digital twinning for healthcare has really become established is cardiology, where it's increasingly finding utility in everyday management of patients. It's perhaps worth just wondering why cardiac modeling has been so successful. People have been working on it for a long time, and very smart people have been working on it for a long time. So that has a lot to do with it. They have great data, and a lot of it is functional and a lot of it is real time. And of course, that helps. But they've really delivered the promise of multi-scale modeling that goes all the way from ion channels through to the hemodynamics of the pump. And in parallel with the development of the cardiac modeling, there's been a whole wealth of activity that kind of hangs around the Physiome project and the virtual physiological human. And this work has been running for over 2 decades now and has generated a whole series of standards. including a systems biology markup language, but also CellML, so a markup language for modeling cells, and FieldML is a spatial modeling framework. But there's also been the production of model repositories that you can just dip into and play with the models. And in many instances, the hosts of the repositories have done a great job of curating the models to make sure that they actually do produce what the papers say they produce. This sometimes includes adding physical units, and we'll come back to that. They've put a lot of effort into building frameworks for modeling and open source software that we can all play with. And if there's one paper that anyone who's interested in doing this stuff, I think you should read, it's this one. The first author is Peter Hunter, who's a bit of a guru in this area. For a long time, he's directed the Auckland Bioengineering Institute. And in this paper, he and colleagues summarize the state of the art in terms of physiome standards and the building of multi-scale models. It's quite a tour de force and comes highly recommended. Just to pull out one fragment of it, they go through the hierarchy of models for complex physiology, and the lowest level is at the protein level, so that's 1 over here on the left. BG stands for bond graph, which I'm going to talk about in a moment. And you'll see that at the top of that list is metabolic enzymes, and immediately underneath that, SLC transporters and ATPase pumps. So those come together in what they describe as functional cell units. And again, you can see that metabolism's at the top. So metabolism kind of runs front and center through all of this. This is an example of OpenCore, which is the Auckland CellML editor and simulation package. You can see in the window, it's linked to the model repository. So you can just click on a model and it'll load up and you can run simulations. On the right is a bit of a metabolism model. And there's just 2 features I'd like to draw to your attention. One is you really have to be very careful with typing the units for every parameter. It doesn't let you save it as a CellML file if you don't get it right. And the other thing which I think is really helpful is that the code for a formula is sometimes a little bit difficult to decipher, but it immediately represents it in a more typical mathematical format, which means debugging is much easier. Another recent publication has just come out, which is a really helpful guide to how to do these models well. And again, it's a good read. And I think that the credibility and understanding goals are critical, but for stuff to be usable, it needs to be reproducible and extensible. So that's a bit of context, but now let's perhaps dive into a little bit of nitty-gritty. If we want to do computational modeling that's mechanism-based, hanging it around physics is a pretty good place to start. And central to physics is energetics. And so this will be familiar to everyone, but just to recapitulate, the things that can happen to energy, it can be stored statically. And for us, quantity of a metabolite is energy storage. It can be transformed. So it can be transformed between electrical energy, or chemical energy in a mitochondria, for instance. It can be transmitted. So for us, that would be down reaction flux. Storage dynamically relates to inertia and inductance, not so relevant for us. Also relevant to us, but sometimes forgotten, is that energy can be dissipated as heat. And of course, at the back of the eye, that may be quite an issue. So this is the most technical I'm going to get into, and this whole subject would justify a series of lectures, so it's going to be very brief. But articles by Peter Hunter, Gawthrop, Crampin, and Pan do a great job of showing how this kind of formalism can be applied to metabolic models. And bond graphs were developed by physicists, largely to make it possible to map energy flows through systems. And the key unit of a bond graph is the bond, which is represented classically, but I think a bit clumsily, by this harpoon. And the key thing about the bond graph, or the bond, is it shows transmission of power. And power is the product of potential or effort and flux or flow. So embedded in the bond are the paired concepts of potential and flux. And the networks come to nodes where there's conservation of power, and there are two types of node. One where the potentials are equal and the flux is sum to zero, and one where the opposite holds true. Now, going into the detail of this isn't feasible in the time available, but I mentioned units being really important, and it really helps understand all this if we break the units down into their kind of fundamental nuts and bolts. And so the forces or potentials that we might be interested in include mechanical forces, so joules per meter or per meter cubed, if we were looking at pressure or flow. Electromagnetic forces, joules per coulomb, otherwise known as volts, and for metabolism, joules per mole. And then there are parallel units for flux. And the issue is that if we multiply those units together, we get joules per second. So that's power. And when trying to get one's head around this, resorting to unpicking the units often really helps. So chemical potential is what we're interested in. The equation at the top is the Boltzmann relationship, which shows how chemical potential relates to the molar fraction of a particular species, Q, over the total present in the solvent. And the flux through a reaction can be determined by looking at essentially the difference in the potential moving the reaction in the forward direction minus the potential pushing it backwards in the reverse direction. And Peter Hunter, in this other paper, which I strongly recommend, has gone through the rudiments of how you get to rate laws for metabolic reactions, starting from the fundamental principles of bond graphs and the maintenance or the conservation of energy and mass or charge. And what's really nice about this is that you can then begin to assemble models that you know are thermodynamically constrained. I'm happy to talk offline about this to anyone afterwards, but we need to crack on. So the reason for burdening you with this bond graph approach is as I say, it helps ensure thermodynamic plausibility. Critically, it allows us to combine models that are either mechanical, chemical, or electromagnetic. And coming to building complex models, it lends itself to creating modular models that you combine by simply connecting them through ports that transmit power from one model to another. So for this bit, I've gone quickly through bits around multi-scale computational biology, talked about some of the tools, talked about bond graphs, and mentioned cardiac modeling has been successful built on data. But of course, in eye research, we're also incredibly
it was not consistent across our samples.
Largely, or in no small part, because of work done by people in this group. We've got fantastic imaging data, whether it's clinical or histology or EM. Great resources in terms of experimental studies and clinical studies. And part of the fun, I think, with building these models is that it provides an opportunity to integrate all of these data in ways that would be really difficult to do so otherwise. What we perhaps don't have that the cardiologists have is quite such good functional real-time data. Then turning to models that relate to the eye, there is, and this isn't by any manner of means a comprehensive list, there is a remarkable richness of computational models in the literature. They're generally rather standalone and so not readily connected to each other, often steady state. Sometimes the only thing that's published is the maths, sometimes there's code as well. Sometimes I can't get the code to work, sometimes I can. But we don't have a consistent framework, and it would be fair to say that very, very few of the published models adhere to the kind of physiome frameworks that I alluded to at the beginning. And this rather garish diagram summarizes some of the models that are around relating to the retina, and I'm going to just point to some of these in a little bit more detail in a moment, but just to quickly sort of orient you. Blood flow is relatively easy to model, so they're good models of retinal blood flow. Choroidal blood flow is work in progress. They're models of Brooks transport and even complement activation, albeit in other contexts. We've started trying to model metabolism in a rather simplistic way. And in the literature, they're good models of metabolism we can try and leverage off. Very nice work from Tianro Guo in University of New South Wales, modeling the biophysics of the whole human retina. More detailed models, for instance, from Delurco on phototransduction. And Christina Keeler has been trying to catalogue some of the processes that go on in photoreceptors and RPE, which is important for reasons that I'll come to. And of course, it's not that interesting to just get a model working for one instance of one bit of retina. What we really want to do is to embrace the fact that the parameters for these models will change depending on eccentricity, depending on individual, depending on disease, and so on. So the modeling framework needs to capture that variability. So why am I preoccupied with metabolism? Well, all living systems depend on it, and equilibrium is equivalent to death. So understanding the energy flows driven by chemical potential is just key to working out how all of these things work. And supply of nutrients, and in this context, chemical potential, is probably a key constraint for many biological systems. And I think metabolism provides a unifying framework that we can use for these multi-scale models. So what about metabolism models? Flux balance analysis has been used a lot. We've played with it a bit. It's found remarkable utility for its relative simplicity. And to try and quickly go through the principles for those of you who aren't familiar with it, the flux balance analysis, indeed this holds, I think, for any metabolic model really, starts with a stoichiometric matrix where the metabolites are the rows and the columns are the different reactions. And so if we just take hexokinase as an example, it consumes one molecule of glucose and generates one molecule of glucose 6-phosphate. And in the process, it consumes one ATP and generates one ADP. This is slightly incomplete thinking about it because it will also generate a proton. And that kind of detail is really important if the models aren't going to fall over. And if you then imagine on the right, it's not drawn, a column vector of the flux through each of these reactions, and you then multiply the rows of the matrix by the vector of fluxes, you then get the rate of change of each metabolite. So if we just take glucose and hexokinase, the flux through hexokinase will determine the rate of increase of glucose minus the rate that's being lost through, sorry, no, I've got this muddled up. So the rate of glucose will be increased by the flux through the glucose transporter, and it will be reduced by the rate of flux through hexokinase. And you can see that this is sort of followed through the entire matrix. And if, for instance, one of these reactions required 2 molecules of something, that would be recorded as a -2 or a +2. So hence stoichiometric matrix. Now, if we take the steady state, then the rate of change of each of the metabolite concentrations will be 0. And so if we solve this, we can begin to get a handle on what the flux might be. But unfortunately, this usually has a potentially huge solution space. And so the art of flux balance analysis is to then reduce that solution space to a single answer. And there are a whole load of things that can be done, and usually many of them are done. So you can set physiological bounds to the fluxes, which you can, to a greater or lesser extent, set according to expression data. And so at its simplest, if a protein isn't missing, if a protein is missing, then and the enzyme isn't present, and that's the only enzyme that catalyzes a given reaction, you can ignore that reaction. It's very easy for the solvers to generate futile loops, and there are tricks to get rid of that. And you can also impose thermodynamic feasibility so that infeasible solutions are excluded. But the final element in this is to solve for an objective function, which basically means that you maximize or minimize one of the reactions in the network. That can be something as complicated as biomass. If you create a reaction in your stoichiometric matrix for biomass, typically or often it's ATP production, because that's something of interest. And the the figure on the right is from a recent preprint from John Doyle's group, where he's got a further sophistication to reduce the solution space, but it's quite a nice diagram in that it shows the big solution space as the outer polygon for flux balance analysis. And the arrow is showing how the solver, if you like, pushes towards an optimal solution, which in flux balance analysis in this instance would exceed what's actually happening in biology. And with further refinements, we can get closer to the biology. And I think this principle of modeling by constraint, as opposed to necessarily building models up from small component parts that become deterministic, is going to be increasingly important. So as with CellML and OpenCore, there are lots of tools available. The OpenCobra repository has tools written in MATLAB, Python, and Julia, with particularly in the Cobra toolbox, which is MATLAB, great tutorials. Actually, they're pretty good in Cobra Pi as well. And I'm guessing most people here will have come across these websites, but they're websites of databases of chemical reactions in a whole variety of species that are essentially genome wide. And so, although they are inevitably, they're not completely complete, they're a fantastic resource in terms of building stoichiometric matrices and so on. So a typical flux balance analysis experiment would be kind of diagrammatically shown on the left. You have your metabolic network that's defined by the stoichiometric matrix. And you can, for instance, set oxygen, glucose flux. It could be any carbon fuel. Need to make sure that water can go in or it doesn't work. And you could equally set fluxes for efflux. And then, for instance, maximize the ability of the network with these constraints to generate ATP. And what we did was try and convert this a little bit to an outer retina model. So chora capillaris being the source of or sink of metabolites, a model with its own stoichiometric matrix for the RPE and another for the rod inner segment. Now, I've emphasized enzymes, but actually transporters are really critical to all of this, and this won't work if the transporters aren't duplicated. So we duplicated all the transporters on the apical side. And of course, when it comes to refining these kinds of models, refining the transporters is a key part of it. And we've played with that, but it really doesn't begin to grapple with what's really interesting. And the issue, and I was a bit slow to wake up to this, but I think it'll probably be obvious to people on this call, is that outer retinal metabolism is demand-led and diffusion limited. And so by demand-led, the major driver of glycolysis and oxidative phosphorylation is the supply of ADP. And so getting the supply of ADP right means we need to understand what the tissue is doing that is generating ADP. And so in particular, what are all the ATPases up to? And the other issue is that the vast majority of metabolite exchange is purely by diffusion, which of course makes perfect sense, because why waste energy if you can do it by diffusion gradient? And again, it's demand-led. So the diffusion gradient between the circulation and the tissue gets larger if a metabolite is being consumed at a greater rate and its concentration is dropping. And of course, this is an exquisite problem, not unique, but an exquisite problem at the back of the eye, because we've got these really significant distances. And so if we're thinking in rough terms between chora capillaris and inner end of the inner segment and 50 microns, glucose takes 3.5 seconds on average to diffuse that kind of distance, which in terms of cell metabolism, when a cell can turn over its entire ATP content in a few seconds, becomes kind of an interesting problem. It's not just glucose. We've got oxygen, lactate, and CO2 going in the opposite direction. And from Jim's lab, not quite such a big distance, but we've also got succinate and malate moving backwards and forwards. And so the concentrations of these metabolites becomes really important. And the big flaw from point of view of what we want to do with flux balance analysis is it doesn't care about concentrations. And so what we need to be able to do is to model all of this with kinetic models that include diffusion. And this is very much work in progress. And I wish I could show you the results of this, but they're not ready yet. But what we have done, and this came out end of last year, beginning of this year, was a very rough and ready tentative attempt at getting what we rather grandly want to call an energy budget for the outer retina. And the idea here, as we work from the right, was to try and get a handle on how much ATP rods, cones, and RPE consume in the light and the dark. And this isn't a straightforward thing to do by any manner of means. And Christina Keel did a great job trying to get a handle on this. But I think we're all keen to emphasize this is the beginning of trying to do this, not anything definitive. And then on the other side of the budget, we tried to work out how much ATP could be delivered in both the foveal region and in the perifoveal macula, where the rod density is high. And we use flux balance analysis purely to define the ATP production from measured glucose, oxygen, and lactate fluxes. And so this depended on the Wang study from 97 and from the great stuff coming out of Rob Linson-Meyer's lab. So these were set from experimental data, and the sort of budget is given in this cartoon, and supply is in blue, and the sort of reddy brown is demand. And you can see the way it fell out was supply exceeded demand, but only just, where rod density is high in the dark. So it's kind of ballpark makes sense. And I think the challenge now is to take this forward in a more refined way. So how do we do that? Well, kinetic modeling is clearly the way forward. And just to give you a flavor, there are lots of glycolysis models out there. A lot of them relate to the same original data set. I quite like this one on the left because it includes buffering of the extracellular space and the cytoplasmic compartment, and shows how altering, for instance, lactate in the extracellular space has a major impact on equilibrium for NAD and NADH. This is another really nice study emphasizing that perhaps we don't need to worry about all of the steps, because a lot of the control of glycolytic flux sits, interestingly, again, two transporters, glucose at one end and lactate at the other, with, surprise, surprise, the phosphorylation steps being the key control points. And John Hoyle, again, has looked at this from a theoretical perspective and shown that control of glycolysis is critical if it isn't going to essentially go out of control and start oscillating. And this seems to be a particular issue for pathways where there's a positive feedback component. And of course, glycolysis produces 2 ATPs for each ATP it consumes. But what I really was excited to share with you, even though I haven't managed to read it all yet, is this paper in the Biophysical Journal. I only came across it 2 days ago, but I think it is exactly the kind of metabolic modeling we need to be able to set metabolic models into the tissue context of the outer retina. And I'm not going to be able to do justice to it, but in essence, what they've done is led by looking at demand. So again, this is now a demand-led model, and what they're showing is in panel A, they impose a step rate increase in ATPase activity, and the model shows that glycolysis keeps up with it and produces ATP that matches that. But the rate of production is only part of the story. We need to know that the concentration is maintained, and they've shown that the concentration is reasonably maintained. And not only that, the rate of hydrolysis of ATP is also maintained. Now, all of that can be done, but you can end up with problematic concentrations of intermediate metabolites. And what they've shown here with the continuous lines is that their model fits rather well with the open circles, which are the metabolite concentrations for the different metabolites in glycolysis. And they've also done some experimental work, which the model can also produce. So I haven't tried running the model. It's written in Julia, which is increasingly used for modeling purposes, but I think this is really cool. So I'm worried about time, so I'm going to skip on. I've summarized why metabolism is important and a bit of why we need to focus on getting this sorted in the retina. So I'm going to talk very quickly about blood flow, unless, Dan, you want me to stop now?
I guess if anybody has any questions now and know that they won't be able to sort of, I don't know, ask later, then you should feel free to ask whenever. But I'm happy to keep going.
Okay, I can probably finish in 5 minutes.
Go for it.
Okay, so this is the other bit of signposting I wanted to do. So this is work from Simon Walker-Samuel, colleague at UCL, that came out in Nature Communications last year. And for us, there's some key bits in this. So the first is he's created a software platform that does simulations of the entire retinal circulation. This solves boundary problems because you only need artery in and vein out. But not only has he done that, he's done it in a way that means that he can move between simulation space and clinical space by using OCTA imaging. And I'm going to skip the detail, but the bottom small panels show how he can go from clinical photographs or OCTA imaging or even a fluorescein to his model, or he can take his model and produce a picture that a clinician would recognize. So this has now advanced quite a lot since the paper, but this is going to provide us with a whole retina blood flow model. But we then need the choroid. So Simon's working on the choroid, but this is a bit of a shout out to Musa Zuash's work, which he's been involved in for about a decade now. This shows the topology of the choroid, or the choroidal circulation, which you'll be familiar with. The red and blue dots being where arterioles either insert or venules drain from this bed. And the key message from Musa's early work was blood likes to go from an arteriole to a venule, and it doesn't like to take the long way around, which means that these areas are relatively stagnant. And so although we like to think of choroidal blood flow as being fantastically high, which it is, supply of blood to the chorocapillaris is not uniformly high. And so there are areas where diffusion of nutrients might be impaired. And Musa's recent Journal of Physiology paper, which was the cover on the beginning of my talk, goes into a great deal of detail about that. So chorocapillaris, not quite as straightforward as we might have imagined. Edward Adams has been taking a different approach, using micro CT reconstructions of the chorocapillaris and using a pipe model, and looking at the impact of changes in diabetes, but with a a different approach to Musa. He comes up with similar conclusions. Other people are, of course, doing this kind of stuff, and that's just a pointer to some of the other work that's being done both in normal retina and disease. So how are we going to put all of this together? Well, it's clearly going to be a community effort. We need to use Physiome standards. We need to optimize data availability. Getting data off PDF supplementary material is a bit of a pain. Getting it off an Excel spreadsheet is lovely by comparison. Metabolism is at the heart of all of this, but we need to couple it to blood flow and what the tissue is up to. This is just mentioning there are a couple of groups in the UK that are trying to push some of this forward with workshops and so on, but this is going to be a global effort, I think. And the final thing I wanted to draw to your attention is that there are now software frameworks being developed that will allow us to hang lots of different models on. You may be familiar with the mycoplasma model, but this vivarium framework has been used for E. coli now. I want us to have a vivarium framework for the eye. So in summary, I've talked too much, so I'm going to skip over that and just say thanks. I mean, there are too many people really, but Peter Coveney and Peter Hunter have been mentors of mine through this for many years. Christine Curcio, Jim and his group, Rob and his group, the data have been just pivotal in getting any of these ideas off the ground. Christina Keel and Stella Prins were working on the energy budget, and I've listed there some of the other collaborators. Important to mention there's a growing community of people around this, and important also to mention Dan for running a fantastic seminar series. So with that, thank you very much.
I'm opening the floor. Does anyone want to ask any questions? I have a lot, but I can ask them anytime.
So can anyone else.
I have a million too. Hey, this is Barb.
Hi, Bob.
I didn't see you at Arvo. Were you there?
No, that's why you didn't
Okay.
see me.
There you go. So what I have, I mean, I'm going to start with the first one instead of going through all the ones that I have. But what caught my attention was that you said that supply supersedes demand in the dark in the Rodridge areas.
So that was
So is
a
that
clumsy,
okay? So even if it was a clumsy way of saying it, do you feel that that might be one of the reasons why the Rodridge area is so susceptible to damage in or where dry AMD develops?
so oh golly, there are lots of bits in that. So the first thing to say is when I say our estimate of supply was greater than our estimate of demand. Both were, I'm sure, wrong. And I just don't know the answer to the biological question you're answering. You're asking. I the point has been made by people on this call that maybe the photoreceptor, so maybe the rod inner segment is relatively hypoxic to protect it from damage. Um, what what I'm hopeful of is that there are now models that include reactive oxygen species generation in the mitochondrial models. So we should be able to get to a point where we can begin to answer the important question you just asked with the modeling. I'm just going to say I don't know.
Okay. Fair
Sounds
answer.
Yeah, thank you for the talk. I just had one quick question. When you were talking about how you built up your model, what like does that only include statistical mechanics, or is quantum mechanical modeling also integrated?
Okay, so I'm not a physicist,
Yeah,
so
totally fair.
everything that we've done is kind of macro.
Okay.
Having said that, there is, for instance, a nice model of phototransduction that's beginning to get to grips with the quantum side of things. There's nothing in the modeling framework that would exclude that as an additional smaller scale, if you see what I mean. And certainly, you know, people are doing that kind of work, but it's way out of anything I could grapple with.
Yeah, totally fair. Cool. Thank you so much.
Okay.
Jason.
Hey, Phil, thanks for the talk. Couple of questions. When you're talking about demand, I assume you're mainly meaning photoreceptor demand, because so there's a second sort of question, which is, in certain diseases, photoreceptors die first, including, for example, SDD based AMD. But then in others, like drusen based AMD, the RPE is dying first. We think, at least grossly, is dying before you get all of the ONL collapse. So I'm sort of just trying to understand, what does that mean for who's driving demand in each of those situations. It's so hard for me to imagine, with choroidal blood flow as it is, that the RPE is ever actually starved for ATP, that it's in an energy deficit. I could believe that maybe for photoreceptors because of the barriers, but I'm just curious, as you're doing your modeling, how much there may be a dynamic play of demand from the RPE versus photoreceptors accounting for this differential degeneration.
Yeah, no, great thoughts. So the kind of our attempt at an integrated model included demand by the RPE as well as by excuse me, the inner segment. And actually what we took, and we tried to take into account at least a bit that some of the inner segment is being provided with at least oxygen from the retina. You're right. As soon as the RPE goes, you take a chunk of the demand out, and that then alters the distances. So another reason why we need diffusion distance built into this. Edward Adams, a student who's doing the pipe flow model in the chora capillaris in diabetes, where you get big dropout in chora capillaris, we do get to a state where I think the RPE might be in trouble, but what I sense is a more itty bitty kind of degradation of the chora capillaris network in AMD. I agree with you. Lateral diffusion is important. And the other thing that we haven't included explicitly, but is in the histology, is that the large choroidal vessels, when the chora capillaris collapses, sometimes seem to be a bit closer to the RPE. And so the big choroidal vessels probably are involved in exchange as well. 25 microns isn't, a ridiculous distance for exchange at all. So we need to model the big vessels as well.
And I guess just as the follow-up, I know Jim and Dan have talked about this a lot as well. And I'm sure Rob Linsmeyer has thoughts, but obviously oxygen diffusion versus glucose diffusion may have different rates. So the limitation factor in particular situations, including when the choriocapillaris drops out, may switch. It may be a glucose deficit versus a oxygen deficit, in various situations.
Yeah, and disentangling that's going to be key. Both oxygen and glucose diffuse pretty well, but obviously with glucose you have to take into account the transporters and the space constraints then of the gaps between photoreceptors, for instance, because it has to go between the gaps, whereas glucose can just plow its way straight through. So, yeah.
Rob.
Thanks.
Hi, Rob.
Hi, this is really interesting, and thanks for bringing this all to our attention. I don't really know anything about this whole area, but
you laid the foundation for it.
Well, not the modeling part. I mean, not the kind of stuff you're talking about. But I wanted to complicate your life a little bit because in trying to understand pH in the retina, where we've just written a big review, we realized that probably from Jim's lab and other people, mostly Jim's lab, I think, that you can't actually separate the outer retina from the inner retina, particularly with respect to lactate, because it's not all waste product going across the RPE. In fact, it looks like hardly any of it is. And that's, for oxygen as well, some of it's coming from the retinal circulation for the outer retina. So I don't know, how are you dealing with that?
Well, we're dealing with it by parking it, but we're not ignoring it. So yeah, I'm desperate to get to the stage where we can at least take into account the Muller cell microvilli and begin to explore some of Jim's stuff about lactate or amino acids, dealing with the sort of like the poverty of glycolysis in the Muller cell. Lactate efflux is intriguing. We set it fairly high simply because the AV difference work in the cat suggested it was quite high, but there are all sorts of problems with doing AV differences, and I completely agree. I mean, in fact, one of Peter Hunter's functional cell units is pH control, and so we have to capture that in every compartment eventually. Yeah.
Good luck.
Baby steps.
All right, Jim.
Yeah, Phil, I was wondering if you from your perspective of making these models, and what information you have and what information you don't have. Could you tell us as biochemists and the physiologists in the group what experiments you would most like us to do to help you make these models? And you don't have to do it now, but could you come up with a sort of triage list of what the most important missing information is that we could work on, to supply you to help you build these models?
Well, that's very kind, very thoughtful, and
I didn't say we'll do them, but we'd like to
no,
know what
I
they
get
should
that.
be.
I mean, the first may be quite, well, it's probably all quite hard. What would be really helpful would be cell by cell or even cell compartment by cell compartment proteomics, so that at least we've got exactly the right players in the right amount in the inner segment, in the outer segment, in the RPE, in the Muller cell microvilli. And in that, I would absolutely include proteomics, transporters, because they're so important. There are various data sets out there which we're trying to curate. And that was partly behind my comment of spreadsheets are nice, PDFs in supplementary material are a bit of a challenge. The other thing is, and clearly some methods demand arbitrary units, but where real world units can be embedded in a paper somewhere is really helpful. And so if you get all the way through and everything is fold changes, it's a bit frustrating, because what we need from a modeling point of view is to understand the normal before the abnormal is imposed on top. And so a fold change is interesting from the point of view of the paper, but we could really do a lot with the raw data of the control state that was considered less interesting by the authors of the paper. So that kind of thing would help hugely. And then I think and this is my preoccupation, which I've kind of inflicted, but how, if we want to understand the dynamics of this, how do we measure ATPase activity in a kind of living retina under different perturbations? So not just light and dark, but high circulating glucose, low circulating glucose. The kind of perturbations that we experience as human beings that the retina is capable of dealing with. So that would be pretty cool. But then I suppose the other impossible thing, pretty well, but we're all preoccupied with why the macula, what's going on in these foveal, kind of cluster of foveal cones that look more like rods, versus a bit further out in the fovea versus the periphery. How can we get regional kind of physiology, if you like, in terms of any kind of biochemistry? Are there nano reporters that can be developed? It's trying to get the data that, I'm talking too much as usual, but that's the kind of immediate response to your question.
Nice.
I'm going to take this time, because no one else has raised their hand, to ask a couple questions. And I want to start with saying I will happily be your experimental gopher for some of this. And actually, not all of it seems that, impossible. But one major question that I guess to start things off is, are we, since there are real anatomical constraints to metabolism that you're kind of talking about with choriocapillaris, maybe shape, where the flow deficits might occur and all that, are we, by working in mice, just losing all of the human physiological relevance? Or is the anatomical sort of similarity enough that we can learn from the mouse mouse still.
So there are two issues there to deal with as quickly as I can. So the first is I think mammalian retina is a mammalian retina, and so I think there's huge value in mouse experiments. First thing to say, and if we got all the rod stuff sorted out, that's brilliant. So and it made you think about cones slightly differently. The second thing, and I think, Rob, we've spoken about this in the past. Metabolism is really interesting, the way it scales between species of different body weights and different lifespans. And I think we could do a bit smarter about making sure that we scale rodent metabolism of the retina to human metabolism when we're doing the comparison. But there's a vast amount of biology that can be gleaned from rodents, and I wouldn't exclude zebrafish or anything really.
Okay. Another question I want to ask, which is especially great since Rob is here to correct me in case I'm wrong. I remember a paper some time back where he supplied different concentrations of oxygen, inspired oxygen, and looked at oxygen consumption. And it looked like at least sort of by eye, it looked like the rate of oxygen consumption was increasing when you supplied more inspired oxygen in the outer retina. And if that is true, to me, this implies that metabolism in the outer retina is not in fact demand limited, but supply limited. Supply being supply of oxygen.
Okay, great.
That's not you. Oh, sorry, that was Yu's group.
So
you
that's
don't
you and
think
Kringle.
Rob does. You never done that in like a cat or anything? Okay.
We have, but we did not actually find a difference.
Okay.
So yeah. Let me not get into the details there.
And maybe I need to be so the metabolism of the rod inner segment, let's be specific, could be demand led but still supply limited. So to be more explicit about that, let's so thought experiment, rod in the day, from Rob's work and others, the PO2 of the inner aspect of the inner segment isn't in its boots. So and so that seems to be working okay. If we then was to stress the rod by putting it in the dark, the PO2 at the inner end of the inner segment could drop to zero and might indeed be diffusion limited, but or supply limited. But the reason it's supply limited is because the demand is so great.
Well, yeah. So if it's normally supply limited, then increasing demand will have a lesser effect on metabolism. And we know from Rob's work in light and dark, at least, that you can increase demand and you can have more metabolism happening as a result. So yeah, I get where you're coming from with this. And it's kind of like some of Rabinowitz's work where he's showing 4 key steps, not just one step, are controlling the rate of glycolysis. Rate control is shared across multiple enzymatic steps. Or in this case, maybe supply and demand are both controlling rate.
Yeah.
Okay. Thank you. And okay, people, please raise your hand if you have questions, because I'm just going to keep asking. Okay, Barb, you should go first.
Thank you. Couldn't find my icon. So I haven't actually read Musa's paper, and I really need to. So that stagnation area of blood flow, what does it correlate with as far as biomarkers is concerned? Because it's in the Sattler layer, right? Which is where all of Jerry's mast cells are hanging out.
It's in the chora capillaris.
It is in the chora. Okay, is it where the chora the capillaries the capillars are? Those pillars between? That is where everybody finds all the complement components pile up, right?
Well, so back to Jim's question, spatial transcriptomics, which I know some folk are up to now, I think it's going to be really interesting because I don't think I think Moose is right. Well, I would do. He's a mate, but and I think the early Hay experiments with fluorescein and primate eyes showing rapid filling of the functional lobule with then a rim around the outside where the dye takes time to clear. I'm sure there's heterogeneity of metabolism at the lobular scale. And spatial transcriptomics is, I hope, going to address that and probably show that the coupling role of the RPE differs depending on where in the chora capillaris lobule you are. So I think there's likely to be heterogeneity of RPE metabolism, but that's
because I was at a dry AMD meeting, and Simon Clark basically said if anybody can figure out what these intercapillary pillars are there for and why they're accumulating so much complement inhibitors, is that the reason why they're there? Because there's this stagnation of blood flow, so there's stress in that area, and then you pile up complement inhibitors to make sure that you don't get too much activation in that area. Is that one of the thoughts?
I haven't been entirely clear. So the functional lobule is say 300 microns across. The pillars, there are lots of pillars within each chora capillaris lobule.
Okay.
So what I'm suggesting would be that the amount of complement, well, the hypothesis is that the amount of complement accumulating in the region of a pillar would be higher at the interface between two chora capillaris lobules and lower in the center where you've got arterial
lots of flow. Yeah. Okay. Yeah, I don't know whether they've actually looked structurally or whether it's all of them or some of them and where they are that have a lot of them.
I think if you just look at a transverse section, there'll be a bit of variation, and actually setting it in the context of the plane of the chora capillaris can't be done. So it requires whole mounts probably.
Right, rather than sections, yep.
And spatial transcriptomics or maybe some clever immunohistochemistry would pick up.
But most people, if they are doing spatial transcriptomics, they're still going to do a whole eyeball section. So they're not going to do flat mounts, they're just going to do transverse sections because that is what we can get to, right? It's really hard to get to particular layers within an eye.
I spoke with one group about this, and the kind of fantasy was to try and take a 2D section where we could then independently reconstruct what's happening in 3D. So at least the 2D has a 3D context, but remains 2D.
Got it. Thank you.
But yeah, it's a challenge.
Jason.
I guess this is follow up to what Barb was asking, and since Rob Mullins is on as well, I don't know if, not to take from Phil's talk, but Rob's on, we could ask both. It's helpful to hear that the lobule size is 300 microns. That's new data that I should have known, but I didn't know. I'm curious whether or not the lobules are reproducibly with relationship to the foveal center point. Is there a reproducibility to the lobular relationship to the foveal center point, or is it random?
Yeah, great question. And So I don't think Moose is on the call. I think he's in a meeting. But so Moose should answer this. Three things to say. One is lobule size varies with eccentricity, and it varies with and its shape. So the shape of lobules changes with eccentricity. Experimentally, but there's a graduate student I'm trying to help who I think may have nailed this. It's quite difficult to do a whole mount of the retina and a whole mount of the chora capillaris and make sure that you keep them aligned, because they tend to come apart. And so I don't know of good data that position the very center of the fovea in relation to a kind of spatial consideration of a chora capillaris lobule. Maybe someone's done it, but I'm not aware. Needs to be done.
And the lobular size gets larger as you get more eccentric. Is that correct?
Yeah.
Yep. Rob, you're on mute if you were talking.
I was muttering to myself here, Jason. Yeah, it also the chora capillaris becomes less lobular the further you get out, and becomes more of a sort of standard capillary bed in the far periphery.
Yeah, it becomes more tubular, doesn't it, towards the periphery?
It's, of course, I just stating I'm sure what all of us would be thinking. If there's a generally reliable relationship between the foveal center and the central lobules, and for example, the central lobule tends to be centered under the foveal center, then you have a susceptibility ring almost exactly, in the location where where we know there's the biggest susceptibility in AMD, the highest rod concentration, 150 microns out from the foveal center or so, which is lines up almost precisely with the most susceptible regions in AMD.
There's other spooky stuff. So Vass has shown, hasn't he, with OCTA, that it's chora capillaris seems to go with aging in that very central bit. What's that about, Rob? So yeah, it's there's lots to learn.
Back a little bit to the modeling. I think in one of the models you spoke about using sort of ATP maximization as the model goals. And I guess my question is that really appropriate, or is there some way that we need to factor in, the change in biomass that, say, rods would need to deal with every day? Or is that insignificant relative to, ATP demands, which probably it is to some extent, but it requires different building blocks. So
yeah, so I had an extra slide, but I won't dig it out because of time. Just doing flux balance analysis and optimizing ATP production isn't enough, is the first thing to say. It's not even enough in terms of thinking about ATP for, in fact, reasons we've discussed, really. So it needs to be set in the context of demand. And you're absolutely right. A lot of ATPases, they're not just about burning ATP. They do stuff which requires metabolites from the circulation. And so as well as having the ATP in our sites, we need to be doing the more comprehensive central carbon metabolism that captures the pentose phosphate pathway, or and yeah, that's all up for grabs.
Rob, you have a question?
Yeah, so Phil, you had mentioned temperature, and it's one of those things that sort of circles back around, like Halley's Comet every once in a while to think about. It's obviously very important, and because all of these reactions and everything that you discussed are going to be affected by temperature and enzyme activities and everything. So there's this paper in the 1990s by Harvard in which he raised the intraocular pressure such that choroidal blood flow was arrested, and was able to monitor an increase in temperature in the retina, just after a few minutes of closing off the choroidal blood flow. So there's this a natural inclination to get hot in the retina if it's not being actively cooled. And but then as you're talking, it occurred to me, it's like, well, this also, not only would this affect all these parameters, but also it's probably affected itself by the rod density in these different regions, and where you have more photoreceptors cranking out, using more energy, especially in the dark. It's probably getting hotter, in those regions, and I don't know. The whole thing's very very baffling, but it's that's just one more dimension that I don't usually think about when I'm considering these things.
Yeah, no, you're absolutely right. And to my frustration, my physics isn't quite good enough. But a back of the envelope calculation, a single rod increases its temperature by 7/100ths of a degree Celsius in a second, which maybe doesn't sound very much, but actually that heat has to be dissipated into the choroidal circulation. So I can't tell you whether it's a critical element in this, but I mean, the other thing, it's quite hard to measure the temperature of a mitochondria, but some estimates put it as high as 40 degrees. And you begin to think, how are they working? Because we think protein should fall over. So, but they're kind of very strange experiments to do. But yeah, temperature's interesting.
And that is in what condition does that rod go up by 7/100th of a degree?
That's in the dark.
In the dark. Okay,
Yeah.
with all the calcium inflow and ATP consumption.
Yeah, all I did was work out the heat of hydrolysis of ATP at the rate required for a rod.
Okay, so I have then a complicating factor of inefficiency that, like, how do you, metabolic inefficiency increases sort of the flux through ATP hydrolysis, increase temperature faster. And there is at least some data, which is not to say that it's very strong data, implying that, say the retina mitochondria aren't extremely efficient at producing ATP. If you inhibit their mitochondrial ATPase with oligomycin, they're still consuming oxygen, perhaps suggesting a leaky membrane.
Yeah.
So yeah, I mean, are you accounting for this in your models, or I assume there must be a good way to account for them?
So we haven't accounted for them yet. It's on the to-do list. Dan Beard and others have produced some really nice mitochondrial models, some of which, in fact, going back to a previous question, do include quantum transition states for the electron transport chain. And they include estimates of membrane permeability, so you can dial in degrees of decoupling, uncoupling, and they include Ross generation. So part of the ambition of this kind of broader brush kinetic model is to incorporate mitochondria and be able to do the kind of things that you're talking about. Absolutely.
Does anybody else have any questions? I know we've taken up 20 extra minutes of some people's time, but I mean, this is super cool. I love talking about this kind of stuff, and I will probably follow up. If nobody has any more questions, thank you so much, Phil, for presenting. This was great, and look forward to talking to you more.
Yeah, well, look forward to engaging with everyone. Yes, thanks everyone.
Thanks, Phil. Thank you.
Thanks for the discussion. It's been great.